Lecture of Unit 3 today. It's going to be on host defenses. There will be 3 lines of host defenses that we're going to discuss. The first 2 lines of defense are part of Chapter 14, so we'll cover the first and the second line of defense along with an introduction to the immune system in Chapter 14, and then our last part of the lecture will be the third line of defense, which is a specific immune response involving antibodies. So before we begin our lecture, make sure you all have everything you need. In addition to potentially your lecture slides, you'll also want to have your course pack notes, and we're going to be filling in notes from pages 73 to pages 78 in our course pack. So we're going to start with an overview of the 3 lines of host defense. So host would include the human being infected. So from our perspective, we would be the host if we are infected. And the first line of defense is going to be a system of barriers that keeps microbes out of the body. So these barriers can be physical, like of the skin. They can be chemical, like the acidity in our stomach, or these barriers can even be biological, such as the microbes that inhabit our gastrointestinal tract. Our second line of defense is going to involve a cellular or chemical response that will involve the immune system. But it's going to include things like inflammation, fever, etc. So it's a more rudimentary form of immunity compared to antibodies. So we'll cover some examples of the second line of defense. Again, some immune system and chemicals are involved, but it's going to be non-specific, which means that these blood cells will attack anything that enters the body that seems to be foreign. And the word innate means we are born with it. So these cells of our second line defenses are cells that we are born with. So what the first 2 lines have in common, the first and the second line of defense, what they have in common is that both lines of defense are non-specific, meaning not targeting any certain pathogen. The third line of host defense will be that specific defense. And so the third line of defense is an immune response, but it's an immune response that becomes developed after we are born. And so it is acquired. We'll also see that being called adaptive. It's an adaptive or acquired immune response. And it's specific, meaning it does target a specific pathogen that's causing a problem, and it usually does that through antibody detection. So what the second and the third line of defense have in common is that these 2 systems involve an immune response. Okay. The first line of defense is what we're going to start with. I've just got this one slide on it, but we're going to talk about it for quite some time. So we're going to go through those physical, chemical, and biological barriers. The physical barrier, the primary physical barrier, is our skin. So skin has an insoluble protein in it called keratin. Keratin is very abundant in the uppermost layers of our skin. And it's very insoluble, which means stuff can't get through it. So if someone with coronavirus coughs on your hand, you would have to lick your hand or breathe in deeply from your hand or rub your hand on your face if you were to get that infection. The virus itself can't be absorbed through the skin because it's impermeable to microorganisms because of this keratin protein. So the skin is a very important barrier to infection, and it's usually only if the skin is broken that microbes can penetrate the skin layer, so through a cut or a wound of some sort. Other physical barriers similar to the skin is we'll have hair, so we've got nasal hair, hair in the genital region. And hair is an important physical barrier that helps keep microbes from getting in, so it helps to block the entrance of pathogens past that barrier. And then within our bodies and in most of our orifices, we've got usually some sort of flushing mechanism. So with our eyes, we can cry. So crying, salivation would be flushing mechanisms where microbes are being moved out of our eyes and mouth. In our nose, we have nasal secretions or coughing and sneezing. We have earwax, that would also be a flushing mechanism. And we have vomiting, defecation, urination. All of these mechanisms remove fluids from the body. All right. Let's see. All right. We, in addition to flushing mechanisms, will have other mechanisms for removal as well. So in our trachea, we will have cilia that line the trachea and bronchi. And these cilia help move mucus in an upward direction as opposed to down into the lungs. So the cilia that line the trachea and bronchi help move mucus upward and out of the lungs. And similarly, in our gastrointestinal tract, we have a process called peristalsis. And peristalsis is the muscle contractions along our GI tract. And this helps move our GI fluids and the microbes in those fluids along towards the anus. So peristalsis can help keep things moving and therefore prevent things from attaching. The more movement there is, the less attachment there will be. So there are probably more examples, but these are the general ones I can think of that act as a physical, a removal, or a flushing mechanism. And all of these things are barriers that keep microbes from getting into the body. If microbes, let's say, do get past the mouth and they make it into the stomach, this is where several chemical barriers can come into play. So in our stomach, hydrochloric acid is secreted from the cells that line the stomach, and this gives it a very high acidity or a very low pH of around 2 to 3 in the stomach. And that will kill a lot of microbes that enter the stomach. Other chemical barriers will include lysozyme found in our tears and sweat. So lysozyme is a review term. We learned about it in Unit 1. And lysozyme is an enzyme that helps break down peptidoglycan cell walls. And so lysozyme, because it can help break down peptidoglycan cell walls, also helps digest bacteria. So lysozyme is an antibacterial enzyme. Similarly, in our secretions, we may also find a variety of antimicrobial peptides, which sometimes can be abbreviated as AMPs. And antimicrobial peptides are, as the name suggests, they're small peptides. They're not going to be as big as a protein. They'll be about 15 to 20 amino acids in length, rather than a protein will have at least 100 amino acids usually. And they have antimicrobial activity. A fun fact, one interesting fact, is that vitamin D is known to help protect against infection because it regulates or stimulates the expression of genes that code for antimicrobial peptides. And so if you're low on vitamin D, you might not be able to make these antimicrobial peptides as well. So vitamin D is usually stimulated by sunlight, or the production of vitamin D is stimulated by sunlight as well. And so in the wintertime, this is why it's really important to take vitamin D supplements, because if we lack that vitamin D, we might be more likely to get sick. And this is thought to be one reason why we're more likely to get sick in the wintertime, because of that lack of vitamin D synthesis. Okay, sebum is a compound, a chemical, it's a lipidy chemical that is secreted by the sebaceous glands. And so this one is not in your book, but I'm going to list it here anyway. So sebum is secreted by sebaceous glands. Sebaceous glands, and these are often found in the forehead. So our sebaceous glands secrete sebum, a lipidy wax, or no, it's not a wax. It's a lipid or an oil. And when it's fermented, it can provide a mild acidity. And so sebum has a mild acidity, it promotes a mild acidity to our skin, which helps prevent other pathogens from attaching. Similarly, our sweat glands will produce sweat, and salt is a compound that's found in our sweat. And so when that sweat evaporates, it leaves that salt behind. And so only halotolerant microbes will be able to survive that high salt concentration of the skin, and many pathogens will not be able to survive. One example of a halotolerant microbe that can take advantage of those high salt concentrations is Staph aureus. And because staph is halotolerant, it can survive on salty skin longer than other types of pathogens. Our skin and vagina also have a mild acidity because of a compound called lactic acid. There are bacteria known as lactic acid bacteria that will ferment sugars and form lactic acid. This is a weak acid and will provide a mildly acidic environment, between 5.5 and 6 usually. And so acidity, enzymes, antimicrobial peptides, salt, these are all chemical barriers that keep pathogens from either attaching to our skin or getting through our bodies. The biological barrier, the only biological barrier I wanted to mention were the microbes that live in and on the human body. The more good microbes we have attached to our skin and found in our GI tract, the harder it is for pathogens to find those attachment sites. Okay, that's it for the first line of defense, our physical, chemical, and biological barriers. All right, and so if I were to give you an example of anything listed on this slide, you should be able to identify it or name it as a first line of defense mechanism. Okay, before we move on to the second and the third lines of defense, because they both involve cells of the immune system, we're going to do a brief introduction into what the immune system is and what the different types of immune cells or white blood cells are. So some basic definitions. Immunology is the study of immunity or the body's protection against infection. And immunity is that resistance to an infection and the pathogen that causes it. The immune system itself is not actually one body system. Instead, it consists of the lymphatic system, which we'll go over in the next slide, and the circulatory system. Specifically, not all of the circulatory system like the heart, but our white blood cells. So together, the lymphatic system and our white blood cells make up the function of the immune system. So the function is what keeps the immune system together, and that is the circulation of defensive cells, the recognition of foreign cells, and then the destruction of those foreign cells. So we can narrow it down to 3 words, this function of the immune system. We could say its job is to search during the circulation portion, recognize pathogens, and then destroy them. So here is a picture of the lymphatic system in a human and some of the important structures or anatomical structures. The first on your list are the lymphatic vessels. And the lymphatic vessels, these are separate from our circulatory system vessels, the arteries and veins. These are a separate set of vessels that move fluids from our tissues back to the circulatory system. And as the fluid goes through these vessels, it'll pass through lymphatic nodes or tissues. And the fluids will be returned to the circulatory system right before the subclavian vein enters back into the heart. And so by passing through all these lymphatic nodes and tissues, the fluid that re-enters the circulatory system should be sterile. So when the lymph fluid, this is the fluid that travels through our lymphatic vessels, should be sterile prior to its re-entry with the circulatory system. All right, so that lymph fluid is called lymph for short, and this lymph is fluid that comes from our tissues. It enters the lymphatic vessels and then is transported, actually powered by skeletal muscle. Our movement helps power the movement of our lymphatic fluid, and so that's why if you move a lot, so movement, or raising your legs will help with the movement of that lymphatic fluid through our vessels. Okay. And then the lymph nodes. So we've covered lymphatic vessels, we've covered lymph or lymph fluid, and the lymph nodes are nodules that are packed with white blood cells or B cells, and the fluids that pass through these lymph nodes will be subject to a lot of immune action. And so the lymph nodes will be where a lot of the microorganisms and pathogens are going to be filtered out prior to that lymph returning to the circulatory system. And it's not just the nodes. So for example, if you feel your neck, you will feel little nodules right there. Those are lymph nodes. We also have lymph nodes in our groin and our armpits and around our knees as well. The gastrointestinal tract, because there are so many microbes in the GI tract, there's also going to be lymphatic tissues surrounding our GI tract. And so lymphatic tissues, or LTs for short, could be associated with the gut. So GALT stands for gut-associated lymphatic tissue. The picture over here says MALT is mucosa-associated lymphatic tissue. That's just a synonym for GALT. and then salt would be skin-associated lymphatic tissue. Similar to the lymph nodes and the lymphatic tissues, we also have an important immune organ called the spleen. And the spleen is adjacent to the stomach. It'll be just above and the left of the stomach. So if you feel the upper left part of your abdomen, you would feel your spleen if it's swollen. And so the bloodstream can carry pathogens if they haven't been cleared by the lymphatic system. So for example, if you have mononucleosis, you'll have an enlarged spleen. And so one thing doctors will look for to diagnose mono would be that enlarged spleen. And so the spleen is similar to the lymph nodes, except instead of filtering out pathogens from the lymphatic fluid, the spleen will filter out pathogens from the bloodstream. And then the only thing I did not yet discuss is the thymus gland. The thymus gland is the site of T cell maturation. The thymus gland is often very reduced after the first couple of years of life, and so oftentimes the only T cells we'll ever have been developed before we were born or shortly after. Okay, so now we're going to talk about the different types of blood cells in our blood, but first let's talk about what blood is. Blood is the fluid that runs through our circulatory system vessels, which would be our arteries and veins and capillaries. And what blood is composed of two major parts. The first part is the plasma, and the plasma will carry all of our water-soluble nutrients, like glucose, and also certain vitamins or antimicrobial peptides. Anything that's water soluble will be found in the plasma fraction of the blood, or the liquid fraction, the aqueous fraction. Then we have the cellular fraction, which includes three types of blood cells. Those three types of blood cells are the red blood cells, which I'll often abbreviate as RBCs. The fancy name for red blood cells is erythrocytes. And red blood cells bind oxygen. There's a protein called hemoglobin in our blood. Hemoglobin has iron at the center of it, and that iron is what holds oxygen tightly within the red blood cell. So red blood cells will transport oxygen throughout the body. That doesn't have a function in immunity, but it does have a very important function. The two types of cells that have functions in immunity would be the platelets, which are the thrombocytes, and then the white blood cells, which are called leukocytes. And so platelets or thrombocytes are involved in blood clotting. Leukocytes or white blood cells, which I often abbreviate as WBCs for white blood cells, these are going to be the primary cells of the immune system. And there are several different types of white blood cells. So in our next slide, we'll go over all of those different types of leukocytes. All right. The first three blood cells, types of white blood cells, are called granulocytes. We have our neutrophils, basophils, and eosinophils. And they're called granulocytes because if you look at them under the microscope, they'll often have this really grainy appearance to them. They'll have a lot of dots on the inside. And these are going to be the most abundant types of white blood cells that you'll see in a blood smear. Any type of white blood cell that is phagocytic or a phagocyte, so a phagocyte is a white blood cell that does phagocytosis. If there's a cell that's a phagocyte, we're going to give it a star. And so neutrophils are phagocytic. They're going to be involved in the second line of defense. So neutrophils are phagocytic. Basophils and eosinophils, these are not phagocytic, but instead they secrete chemicals and they respond to allergies, and they're associated with inflammation. Eosinophils, side note, are also involved in killing worms. If you have a parasitic worm infection, ew, then eosinophils will be elevated. So you can tell a lot from a blood count or a blood smear. If you do the different types of cell countings, if neutrophils are elevated, that means you probably have some sort of infection. If your basophils or eosinophils are elevated, that means you probably have some sort of inflammatory event. If all three are elevated, that means it's probably inflammation due to an infection. If only the basophils and eosinophils are elevated but the neutrophils are not, that could suggest some sort of autoimmune cause for an inflammation. Our monocytes include macrophages and dendritic cells. These are going to be very large phagocytic cells. So our monocytes are also phagocytic. And we're going to see more of these. We'll learn more about these in our third line of defense. Our lymphocytes are not phagocytic. Instead, they produce substances that harm pathogen, like B cells produce antibodies, and antibodies will target pathogens for destruction, but they won't actually destroy or engulf the pathogen themselves. And then our T cells, there's going to be two types. We've got our helper T cells, or our TH cells, and then we have our cytotoxic, or our killer T cells. And our helper T cells are going to stimulate B cells. And then our cytotoxic T cells will destroy or damage our own cells that are either infected or damaged for some other reason. So we'll talk about neutrophils more. We're going to talk about these more, and we'll also talk about B cells and T cells a little bit more. Okay, so for our second line of defense, these are going to be our nonspecific and innate immune responses. And so there will be three examples that we will go through. The first example is going to be phagocytosis by neutrophils or other phagocytes, so perhaps macrophages or dendritic cells. But we're going to focus mostly on neutrophils here. So these are the primary phagocytes of that second line of defense. So let's define what phagocytosis is. Phagocytosis is when a white blood cell, or a phagocyte, because remember, not all white blood cells are phagocytes, will recognize a bacterium it'll attach to the microbe, after it attaches to the microbe, it'll then ingest the microbe through phagocytosis, or it'll engulf it. And then once the pathogen is engulfed, the pathogen will be digested. So a white blood cell will recognize the pathogen, attach to the pathogen, engulf the pathogen, and then digest it. So your book goes into a lot more about how this works. I like to keep it simple by just drawing my picture of a white blood cell as a Pac-Man eating a bacterial cell. And then once that bacterial cell is ingested, it'll fuse with another cellular component called a lysosome. The lysosome will fuse with the ingested particle, and then it'll digest that bacterium within the white blood cell. And the phagocytic debris would be the leftover digested material from that pathogen that was ingested. Gross. So the one term your book talks a lot about, and this is not going to be in your test, but it is important to understand if you do the reading. There's this process called opsonization. And this is when the white blood cell coats the bacterium or the pathogen with opsonins or proteins or small molecules. It'll coat the pathogen with small molecules that it will then be able to recognize and use for attachment. So opsonization coats the pathogen with molecules so that it can be recognized and attached and then engulfed. So this opsonization makes attachment possible and engulfment possible. One of the reasons I'm bringing this up is because in our last lecture we learned that capsules could help cells evade phagocytosis. And the reason this is because a capsule prevents that opsonization process. And because, well, hold on, I'll repeat that. So capsules can protect the cell from opsonization. And therefore, it prevents the cell from being, the bacterial cell, from engulfment by a white blood cell or by a phagocyte. Here's a video that will visualize that for you a little bit better than I could.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
All right. And so you all won't be responsible for knowing the detailed mechanism of phagocytosis. You really should just know what phagocytosis is, what phagocytes are, and that phagocytes, like neutrophils, are a second line of defense mechanism for host defense. Another important defense mechanism in the second line is inflammation. So inflammation is also going to involve phagocytosis, but it also involves a lot more than that. All right, so inflammation usually happens when a tissue is injured. So if this is the surface of your skin and you get a splinter, then microbes will get in to your skin because of that injection. And within our skin, we have blood vessels, and the blood vessels will immediately constrict, and then they will dilate again. And fluids will enter the scene. So once these blood vessels dilate, they kind of open up, and fluids from our blood can enter the scene. And this is where our neutrophils come in, or our white blood cells. Once the neutrophils come in, They start gobbling up the microbes, and the fluid will also accumulate from our bloodstream, and that's what causes that swelling, which is called edema. So edema, that fluid accumulation, and then phagocytosis, which is neutrophil action, is then followed by pus formation. So I've got a fun definition for you. Pus is a combination of white blood cells, bacteria, and gross phagocytic debris, which means after the neutrophils do their thing, the phagocytic debris is the digested components of that bacteria that have been expelled. Once the neutrophils have done their job, the tissue can then be repaired. Tissues can return to their normal condition, but oftentimes scar tissue will build up. So tissue repair will involve the formation of scar tissue, and this can do a lot of damage. So chronic inflammation can form a lot of scar tissue, and this is why chronic inflammation can be damaging. All right, here's another video to show you a visualization of what we had just talked about. And kind of like with phagocytosis, you're not going to be responsible for knowing all of the mechanisms involved in inflammation, just knowing what inflammation is and that it's a second line of host defense.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
All right, so that also showed you a good image of phagocytosis there at the end, where the neutrophil would move toward the bacteria via chemotaxis. It would attach to the bacterium and then engulf and digest it. So inflammation also involves phagocytosis, and both inflammation and phagocytosis by neutrophils would be considered second-line defense mechanisms. Our last second-line defense mechanism that I'd like to talk about is fever. So fever is our third example, and fever is an elevated body temperature. A temperature above 98.6 is usually considered a fever. 98.6 tends to be the normal body temperature. That could be a bit of a high estimate. Normal body temperatures can be anywhere from 97 to 97.5 to 98.6. So fever is just elevated body temperature. Whatever your normal body temperature is on a daily basis, if it's elevated by more than a half of a degree or a degree, then it would be considered fever. All right, so fever is an important thing protect against infection. So bacteria need iron in order to grow and divide, and the heightened temperatures can limit iron availability. That can also make us very tired, but it'll also lower bacterial growth rates. The temperature itself, because it'll be just outside of the optimal temperature for bacteria, that elevated or slightly higher temperature can have a negative impact on bacterial growth rates. Fever can interfere with viral replication. It can increase the effectiveness of the inflammatory response or our other second-line defense mechanisms, and also phagocytosis. And it can stimulate the production of other immune molecules, like those opsonins that will coat cells. So a lot of these things are related to one another, but fever does help protect against infection. It's usually one of the earliest signs of an infection reaching its stage of causing damage. But while moderate fevers can be a good thing, low to moderate fevers are help with the above. High to extremely high fevers can be dangerous. And so if you have generally a fever of above 104, this would be a reason to seek medical attention. And a fever above 106 would be extremely high and very dangerous. So an extremely high fever can result in death. So everything is good in moderation, including fevers. All right. So now we're going to move on to our third line of defense. Our third line of defense is specific, and it is acquired or adaptive. Here's our definition of immunity again. Immunity is that long-lasting or acquired resistance to an infection, and it's usually due to some sort of previous exposure. We've got a couple of terms, and to tell you the truth, I don't really care if you memorize humoral versus cell-mediated immunity. Humoral means it's associated with the fluid. To put it simply, humoral immunity is going to involve our B cells and antibodies. And cell-mediated immunity means that one of our cells is affecting another one of our cells. And so our helper T cells affect B cells, our killer T cells affect our host cells that are damaged and infected. So our cell-mediated immunity will involve T cells and their response. All right. So acquired, again, this just means we are going to build or receive it at some point after we are born. And this process can be natural. So a natural way to build or acquire immunity would be to get sick. An artificial way to build or receive immunity would be through a vaccination. All right, we can receive immunity naturally through breast milk, for example, or we can receive immunity artificially through a serum donation, where you get plasma donated from another individual that hopefully has the antibodies in that plasma to fight the infection that you have. So we'll go more over this again. And immunity is specific to the one pathogen causing the disease. It's going to involve recognition of antigens. And the B cells will form memory cells, which will have a long-lasting memory. And so this means you're protected from infection after you've developed the response. By the same microbe. So some other general terms that we're going to go through are going to be antibodies and antigens. We'll start with antigens. Antigens are small molecules. They can be peptides or carbohydrates or a combination of the two called glycopeptides. Regardless, they're small molecules on the pathogen's surface. So this surface could be the cell envelope, like the cell wall, or the outer membrane, which again is found only in gram-negative bacteria, or antigens can be found on the flagella. So this would be a flagellum. So we've got these little molecules on the surface of our cells that can be recognized by antibodies, which are produced by human cells, by our white blood cells. So antibodies are small peptides produced by B cells that recognize and attach to antigens. All right, and these antibodies are also, a synonym for antibody would be an immunoglobulin. I usually say antibody because immunoglobulin is a mouthful. All right, and so antibodies will do several things to a pathogen. One thing they do is they mark it for destruction by a phagocyte. So macrophages, for example, will recognize antibody-marked pathogens and then engulf them. So kind of like opsonization with the opsonins, antibodies can also act as opsonins so that macrophages can attach to the pathogen and engulf them. So an antibody-marked pathogen is going to be much more likely to undergo phagocytosis than an unmarked pathogen. Antibodies can also have other important roles. So if antibodies are attached to bacterial flagella, that will inhibit movement, and it can also prevent the adherence of that pathogen to our host tissues. And so adherence is one of the important stages of causing infection, and movement can also be critical in the human body as well. So if bacteria are unable to move, they may be more likely to be hunted down by our white blood cells. Antibodies can also neutralize viruses by attaching to them and therefore preventing them from entering host cells. And antibodies can also neutralize toxins as well by kind of blocking them up. If a toxin is blocked by or neutralized by antibodies, then the toxin won't have the same damaging effect on host tissues. So next we are going to talk about the development of this type of immunity, or in other words, how is this antibody response formed to a specific pathogen? So we're going to go through 4 different cell types that play a role: the dendritic cells, the helper T cells, B cells, and macrophages. And so the picture is drawn right here, and you can draw a little bit of this picture or see a little bit of it in figure 15.1. There's also some space for you to draw it on page 76 in your course pack. So the first thing that happens, or you could just write out the steps, the first thing that happens will involve a dendritic cell. So our dendritic cell is a phagocyte, and it attaches to a pathogen and engulfs it. And here is an antigen on the pathogen surface. So the dendritic cell engulfs and digests the pathogen, and then it takes that antigen and puts it on its own cell surface. It takes that antigen and delivers it to a helper T cell. And so because it's delivering this present to the helper T cell, and because this present is an antigen, we call the dendritic cell an antigen-presenting cell. It's going to engulf the pathogen, incorporate those antigens on its own surface, give the pathogens to another cell. So next to dendritic cell, you can say it's an antigen-presenting cell because it does these things. All right, so the helper T cell, what it does, once it has been activated by receiving this antigen, it will send chemical signals to B cells to start responding. All right. So when the helper T cells send these chemical signals, the B cell becomes activated. When the B cell is activated, it's going to do two things. The first thing that the B cell does is it'll make antibodies for the pathogen. The second thing it'll do is it'll produce a memory cell, which will have that long-lasting recognition of pathogens and their antigens, and therefore it'll be able to destroy pathogens should they ever come back again. So B cells, technically they differentiate into plasma cells, which make antibodies, but to keep it simple, I say that the B cells will make antibodies that will mark our pathogen for destruction. Our macrophages will be the cells that ingest and digest through phagocytosis, engulf our antibody-marked pathogens. And then the B cells also will form those memory cells to recognize the pathogen again in the future. The figure our next example of third line of defense will be our T cells or cell-mediated immunity. We're specifically going to talk mostly about cytotoxic T cells. Again, the helper T cells assisted B cells, so that was how that was cell-mediated. But our killer T cells, or our cytotoxic, toxic is the killing aspect, cyto refers to our own cells. So T cells that are killer T cells will damage our own infected or damaged cells. So a killer T cell usually will produce some sort of toxic substance so that it'll destroy our own cells that are either infected by a virus, infected by a bacterium, or our damaged cells like cancer cells. They can induce apoptosis of those cells. So T cells deliver death package chemicals that will cause an infected or a damaged cell to undergo self-destruction or apoptosis. This is going to summarize a lot of what we've just learned about that third line of host defense.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So one thing I'd like to point out after this video is that one reason a coronavirus infection can be so dangerous is if the T cell response is extremely heightened. So if we have too much of an immune response, this can lead to massive damage of our own cells. So if our lung cells are becoming damaged, that can cause a lot of harmful scar tissue in the lungs, and they may be irreparable. So this can cause massive damage to tissues. Another thing that can happen with an too much of an immune response is we could get a lot of fluid accumulation, or that edema that I was telling you about. And if a lot of fluid accumulates in a tissue like the alveoli of the lungs, this could cause pneumonia, and that is another thing that can result in death. So an overreaction of our immune system is also something that can be harmful to us. I want to end by talking about vaccinations. And we're still in the third line of immune defense, except this is going to be an artificial form of immunity, or third line defense. And so vaccines or immunizations will involve the formation of an antibody response. So again, this acquired immunity, what I mean by that is the formation of an antibody response. So we're still in the third line of defense, right? This, we went over how someone develops that response by having an active infection. That's what we just went over. If you receive antibodies through breast milk, then that would be passive natural immunity. Passive because you're just receiving the antibodies from another person. But vaccines involve forming the antibody response in an artificial way. And so if you are exposed to the pathogen's antigens or a part of the pathogen, then our body will make the antibody response in the same way that we just went over, as if it were an active infection. But because vaccines don't inject you with a live pathogenic form of the pathogen, you cannot get the infection from the vaccine. You're just receiving an inactivated pathogen or a part of the pathogen, its antigens, for example. And so a vaccine will stimulate, it'll mimic the infection, stimulate the appropriate antibody response. So that way you'll have the antibodies in case you ever come down with the real infection. This can protect communities or help prevent infectious diseases from spreading in a community through something called herd immunity. So herd immunity is when a significant proportion of the population, say 60 to 80% of the population, has that antibody response. So right now, our population does not have that antibody response to coronavirus. If we were to develop a vaccine and vaccinate 60 to 80% of individuals, then that would lower the spread of the infection through the community, and that will also help protect the most vulnerable. So the most vulnerable at least with coronavirus, are the elderly, those with underlying chronic health conditions like high blood pressure, diabetes, um, asthma, any type of lung issue um, would make you a high risk individual. And so, if vaccines are available, like for influenza, for measles, for pertussis, then a significant proportion of the population becoming vaccinated can really contribute to this herd immunity. And the people that benefit the most are those that are the most vulnerable from getting sick or the most high risk. So here are some vaccine preventable diseases: influenza, pertussis, which is whooping cough, polio, which is caused by poliovirus and a pathogen of the nervous system, causes paralysis, cholera, typhoid fever. Typhoid fever is caused by Salmonella enteritica, a pathogen of the GI tract. Both cholera and typhoid are caused by bacterial pathogens. And then tuberculosis, measles, mumps, rubella are viral infections that can be prevented through a vaccine. And then chickenpox and also shingles are caused by the varicella zoster virus. So these are just a few examples of vaccine preventable infections. Vaccines can come in an inactivated form, and this means that you are being injected with just a killed virus or pathogen, or just a part of it. So inactivated means it's been killed, or you're receiving just a part of that pathogen. So it's safer, but it can be less effective. And one reason it can be less effective is because you might get it through a shot in your arm, and then therefore it enters through the wrong portal of entry. Right? So influenza would normally enter through the nasal or oral passages, not through the arm. So attenuated vaccines will mimic the actual infection better because it'll go through, if you get like a nasal mist, it'll go through the actual portal of entry that the infection would normally go through. So you're given a live organism, but it's been altered so that it's not pathogenic. It'll result in a better immune response, but it is a little bit less safe. And so anyone that's considered high risk, it's not suggested that you get these attenuated or live vaccines. There's a chance it could mutate, or your body could have an overreaction, or the attenuated strain could, if your immune system is really, really down, it might do damage where it normally wouldn't. So this is not a part of your lecture review guide and not part of your lecture outline, but since you all aren't going to be writing the writing assignment on this, I thought I would do just end quickly with some fears and facts about vaccines. And one of the most common fears is that vaccines might cause autism. And I can tell you all that there's absolutely zero evidence that links vaccines with autism. There was a paper that was published by Andrew Wakefield in, I don't remember what year, I believe it was the 80s. And it was filled with falsified data and claimed that this link occurred. This paper has since been retracted, and no other paper since has linked vaccines with autism. And so this is something that is a fear, not a fact, that vaccines cause autism. They do not. You might be scared that the vaccine is toxic. So there are some preservatives or ingredients in vaccines that keep microbes from growing in the solution. So thimerosal, for example, is a mercury-containing preservative. Some facts about thimerosal: it has been proven to be safe. It does contain mercury, but probably less mercury than you would get from eating fish. And the form of mercury is also a safer form of mercury than that's found in fish. So we wouldn't be getting exposed to something that we're not already exposed to by having this vaccine that has thimerosal in it. And the ingredient thimerosal, so even if it does, freak you out and you don't want to put it in your body, the ingredient thimerosal is not found in the single-dose flu shots that you would normally get from Rite Aid or Walgreens. And then finally, the flu shot itself cannot give you the actual flu. So the vaccination will not cause the illness. You may experience some symptoms like a fever or chills, and what's happening is your second line of defense, that inflammatory response or that fever, is just kicking in. And so you are not actually coming down with a fever or chills in response to a real infection. It's just that normal second line of defense or that normal innate immune response. So if you do come down with a slight fever on the day you get a shot, that just means that your second line defense mechanisms are in order and working well. But that fever should be gone within a day. That inflammation should be gone usually within a day. If you actually do come down with an active case of influenza after you've gotten a flu shot, then one of two things might have happened. Chances are you were exposed to the virus before you got the flu shot. It takes about 10 days to 2 weeks for the flu shot to actually result in that antibody response. So if you were infected with the virus a couple of days before you got the shot, that virus will reach the infective stage before your immune system has time to catch up. So your immune system might not have had time to form that antibody response. So if you get the flu after you get a flu shot, there's a good chance you were exposed to the virus before you got the vaccination. And then finally, the vaccines are only as effective as our antibody response. And so if you're feeling sick or tired, or if you're feeling low or stressed the day you get your shot, then your immune system might be lowered and not have the proper response that it should. So it's always important to get your flu shot on a day that you feel well rested and fairly energetic and healthy. There is always a slight mismatch of, or not always, there's often a mismatch of the strains that are used in the vaccine development and the strain of the influenza that's actually going around. Even if there's a slight mismatch, the flu shot still offers some protection against the influenza no matter what strain is causing it, and you're less likely to have the types of severe complications that will result in death. And so even if you got an imperfect flu shot, it's still better than no flu shot at all. All right, so that's all I have on vaccines. It's a third line of defense. It's an artificial form of active immunity, but it still promotes a natural response to infection by allowing our B cells to make antibodies for the pathogen that's causing the infection. All right, all, that's it for Unit 3. We will have a live Zoom session on Wednesday for questions and answers, and our test will be next week. Good luck.